So the next set of notes has to do with the characteristics involved in what we call the course of infection. Now, the course of infection is something that you've all experienced. You've all been sick, so you all know what it's like to go through this incubation period. You come in contact with somebody that's sick, right? You don't really pay much attention to it. Maybe you shake their hands, maybe you give them a hug, maybe they sneeze right in front of you, okay? You come in contact with the pathogen. From that point until the time you start to get ill, or what we call the onset of illness, is basically called the incubation period, okay? Now, this can last anywhere from a few days or up to 2 weeks, so up to 14 days. And it is determined by the number of organisms encountered, the condition of the host, and many other factors. For example, if there is a high infectious dose number, the incubation period might be shorter. If the person has an immune system that's suppressed, or they're stressed out, or they're not eating right, or getting enough sleep, incubation might be shorter, okay? Following the incubation period, there's what's called the phase of illness. Now, in research, the phase of illness can be divided into 2 categories. Either A, the prodromal stage, or B, the invasion stage, okay? Some research journals will tell you just the phase of illness. And so basically what's happening is this is where the organism is moving into its target tissues and the major symptoms appear, okay? That would be considered the prodromal stage. In the invasion stage, the microbe is multiplicating at a maximum rate, exponentially, in that target tissue, causing disease. So let's say it is a respiratory pathogen. You inhale it through your nasal passages, but it actually targets your lungs or your bronchial tubes. That means it has to move from the nasal passage down the back of the throat into that target tissue in order to cause the illness. So that would be the difference between prodromal and invasion. Now, when we look at it this way, actually I'm kind of getting ahead of myself. The third stage is called convalescence. And this is considered the stage of recuperation and recovery. So basically you're getting better. Now that could occur one of 2 ways, okay? You can either go to the doctor, you can take an antibiotic for bacteria, maybe some antiviral medication, but usually if it's a virus, the doctor's just going to tell you to wait it out, okay? So then your immune system has to fight it off. If your immune system has to fight it off, that's natural immunity. But here's what you need to know. During the convalescence stage, microbes can still be shed, and you may still be contagious even though there are no symptoms or signs of infection anymore. Now, in our last unit, we looked at acute viruses, chronic viruses, and latent viruses. Those types of infections, okay, still fit the bill for following these 3 stages. In all 3 of them, there is an incubation period that usually takes only days. Then there's the period of illness. Now notice how acute is fairly short. And then convalescence occurs. You can, again, go to the doctor, or your body can get rid of it all on its own. Usually, immunity exists after that. In a chronic infection, the person is sick over a long period of time, or the illness recurs frequently. In a latent infection, notice how we go through incubation, illness, convalescence, almost like, wow, I must be getting better. And then there's kind of a latent period where there's no signs, there's no symptoms, and then all of a sudden illness occurs again, okay? It's reactivated. So when we look at the pathogen itself, okay, there's going to be lots of different characteristics about the actual pathogen that we can study in terms of how it infects a host. So first, it could cause what's known as a localized infection. This is where it's limited to a small area. Then we can say it's either systemic or it's generalized, okay? meaning the infectious agent is spread throughout the body. Maybe it gets into the bloodstream or the lymph vessels and then is transported. Okay? Now, here's the thing. Some bacteria can get into the bloodstream. That is typically referred to as bacteremia. Okay? Emia itself means in the blood. So if the bacteria in the bloodstream, that's what we call the infection. One really common example of that is vigorous toothbrushing. If you're using a toothbrush that's too hard and it makes your gums bleed, the bacteria from your mouth can get into the bloodstream, and that can actually make you sick. Okay? They're actually finding a link with that with heart disease, that they're thinking that's how some of the bacteria actually get into the heart. Toxemia would be toxins in the bloodstream. Now, tetanus is a really good example of toxemia. Viremia is when viruses or other viral products are in the bloodstream, and an example of that is HIV. And then there's septicemia. And I've talked about septicemic shock. Let's see if you remember what that's from. It's when an acute life-threatening illness occurs that's caused by infectious agents or their products circulating in the bloodstream. One example is toxic shock syndrome. Girls, you know what I'm talking about. Boys, this is an illness associated in females with the use of feminine products. Okay, so septicemia, infectious agents or their products. Remember me talking about the bacteria that are gram-negative, that have LPS, and LPS acts as one of those infectious agent products. Okay, if it gets in the bloodstream, the person gets septicemic shock. Now, when we look at the establishment of an infection, there are several portals of entry in which colonization occurs. Okay, lots of vocabulary in this unit. We're going to do an activity that's going to help you with all this. But the portals of entry of the human body include skin breaks, so if you get a cut or a scrape, your nose, your mouth, your eyes, your ears, vagina, urethra, and anus. Okay? So we've got this pathogen. It's at the portal of entry. It's trying to colonize. In order for it to colonize, it has to adhere. Okay? So adherence is the process the microbe uses to make the contact and actually attach to the host cell's receptors. Now, some of these should sound familiar. There are adhesion mechanisms that include fimbria, the capsule or glycocalyx, and adhesins, or those little pili, on bacteria. You can see a picture of those little pili right here. Remember, we talked about those. Viruses, they use their protein spikes. Protists then use hooks or filaments. All right? And then this image is just depicting the bacterial cell capsules. Okay? Now, once the pathogen has penetrated the mucous membranes, most of those pathogens have some way to get into the tissue, and somehow they have to travel to their target tissue. So the example I used before was maybe you get a respiratory virus in through your nose, but it's actually attacking your lower respiratory tract. It has to somehow get from point A to point B, from that portal of entry to the target tissue. So there are two different ways here on this page that shows how this happens. So there can be uptake by an epithelial cell. Some pathogens induce cells to take them up via endocytosis. Okay? There's exploitation of the antigen sampling process. Now what that means is it's a microbe that actually wants to get caught, wants to be identified as something foreign so that your white blood cells will engulf it. But then what you maybe don't realize is the white blood cells have a free pass. They can go anywhere they want in the body. So some microbes have developed a way to escape from the white blood cell and then actually infect other cells once the white blood cell gave them a free ride. So one example here is chlamydia. Chlamydia between 48 to 72 will get taken up by a host cell, right? It will actually divide inside of that cell, and within 48 to 72 hours release all these other cells in order to go and infect new ones. So this is just an example over here of a fluorescent tag that was associated with the bacterial cell on the inside of a white blood cell so that you can actually see it. It's kind of cool imagery. Now, one other thing that we talked about in the previous set of notes was virulence. The more adaptations or traits a pathogen has, the better it's going to be at causing disease in a human or host. Remember, this is their whole purpose. Okay? They want to get in and make more, whether they're bacteria, viruses, fungus, it doesn't matter. So one example is going to be that what I just talked about with hiding within a host cell. Okay? One is going to be what we call antiphagocytic factors. If they're using antiphagocytosis, that means they're trying to avoid the white blood cells. So the microbes can either avoid phagocytosis using chemotaxis, so movement, they actually move away from the white blood cell. They use recognition, attachment, and then fusion inside the white blood cell and actually destroys the white blood cell. So this example in this picture down here is showing a white blood cell about to attack this bacteria. And here it's been trying to take it up. But notice how instead of engulfing it, it looks like the pseudopods are actually sliding back off again. It can't get a hold of it because it's got a capsule that's actually giving it the ability to be slimy. Okay? Another way might be those microbes produce membrane-damaging toxins that actually destroy the white blood cell from the inside out. Some, like that chlamydia example, actually multiply in the cytoplasm. So those are some ways to avoid those white blood cells. Now, antibodies are the proteins that your immune system produces that enable you to recognize an infection or a pathogen when you come in contact with it a second time. Some microbes have an enzyme that literally cleaves or cuts part of those antibodies to avoid being recognized. There's antigenic variation. We talked about genetic reassortment. They literally change some of their receptors on their cell membrane to stay ahead of antibody production. Think about the flu and how quickly it mutates. Some pathogens will actually cover their surfaces with receptors that mimic the surface of the host cell so that they resemble self. An example of that would be viruses that take on part of that host cell membrane and create an envelope. Now, another example is going to include or determine how virulent they are is to look at how they damage the host. Okay? So this is going to be effects to the host and host tissues. So in order to cause disease, a pathogen must evoke some kind of damage. So they can release exotoxins. And you can read this about exotoxins, but there are different types or categories. Neurotoxins are toxins that are released to damage the nervous system. Okay? Enterotoxins cause disturbances to the gastrointestinal tract, like severe vomiting and diarrhea. Cytotoxins cause a variety of cell types to be damaged, either by interfering with their cellular mechanisms or literally just flat out lysing the cells and causing them to explode. Okay? An example of a neurotoxin would be tetanus. An example of enterotoxins would be amoebic dysentery. And off the top of my head right now, I can't think of a good example for cytotoxins. So we'll leave that one blank. Some pathogens have what's called phospholipase, an enzyme that literally rips those lipid heads off of the cell membrane, which makes the cell unstable. Some are called superantigens, super foreign material that activate the immune system by stimulating 1 in 5 cells instead of 1 in 10,000 to produce a chemical called cytokine. And cytokine causes symptoms like vomiting and diarrhea, fever, nausea, and even shock. So the major example there would be toxic shock syndrome. Some have endotoxins. Those would be the gram-negative bacteria that have those lipopolysaccharides. Some elicit the inflammatory response and can actually destroy tissue itself by having the white blood cells squeeze out toxins and enzymes that they're supposed to contain. Bacterial meningitis actually does that. So that's some examples of how pathogens are virulent. And now we're ready to move on.